A metabolite is a small molecule that is usually either an intermediate or an end product of metabolism. Some examples of metabolites include acetic acid, ethanol, and glycerol, but there are many more beyond this. Some metabolites act as substrates and therefore stimulate enzymatic reactions involved in metabolism. Because reactions that take place on the cellular level must be very controlled in order to adequately carry out cellular functions, such as glycolysis and the citric acid cycle, the pathways of these metabolites must be carefully regulated at every possible branch point in order for the enzymatic reactions to take place correctly. Specifically, when a cell's ATP stores cannot be replenished due to lack of food for animal cells or sunlight for plant cells, cells experience transient deprivation. To prepare for this, cells can build stores of energy to be used during transient deprivation. Cells must constantly decide whether metabolites should be sent down anabolic pathways to build an energy store or sent down catabolic pathways to create immediate energy.
As mentioned, this process and reaction are intricate and exact. This process of metabolite acting as a substrate to fuel enzymatic reaction is somewhat like organized chaos, as a single substrate can be an integral part of several different pathways that all modify the metabolite in different way. Each pathway complete for that metabolite and with several different pathways competing to modify thousands of different metabolites in different way, some form of organization is necessary. This calls for control mechanism within the cell that regulate and coordinate this complex activity. Control mechanism can include both positive and negative feedback regulation.
To discuss an example of a substrate that adds to the chaos of this process, pyruvate is a metabolite that acts as a substrate for several different enzymes that chemically alter the metabolite in a different way. Pyruvate is part of the pyruvate dehydrogenase complex, which is used to produce acetyl coenzyme A. During this process, lactate dehydrogenase can convert pyruvate to lactate. Another enzyme converts pyruvate to alanine, and another converts it to oxaloacetate. Covalent modification is then used to control how much pyruvate is modified by each enzyme in order to organize the use of pyruvate in anabolic and catabolic processes.